Welcome to Section 9 of Genetics. In this section, we'll be discussing trinucleotide repeat disorders. Let's get started. Trinucleotide repeat disorders are exactly what they sound like. They're caused by an abnormal segment of repeated trinucleotide bases within the genome. We discussed anticipation in Section 1 of this chapter, but you should know that anticipation is common in these disorders. As a brief review, anticipation is the tendency of a disease to occur earlier or to be more severe in succeeding generations. This occurs because these repeated segments in the genome are somewhat unstable and can be significantly expanded in offspring, making the disease more severe. The exact mechanism here isn't very well understood. Okay, there are 4 trinucleotide repeat disorders you need to be familiar with, and they can be remembered with the mnemonic, try hunting for my fragile eggs. Tri stands for trinucleotide repeat disorders. Hunting stands for Huntington disease. 4 stands for Friedreich ataxia. My stands for myotonic dystrophy. And fragile eggs stands for Fragile X syndrome. This is a table of the trinucleotide repeat disorders, which can be found in Section 9 of Genetics. Huntington disease is an autosomal dominant condition caused by CAG repeats. The repeats occur in the HTT gene on chromosome 4 and result in basal ganglia atrophy. Recall from neurology that the basal ganglia is this region of the brain right here. This includes many structures such as the caudate nucleus and lentiform nucleus and is important in regulating movement. In Huntington disease, this part of the brain is affected, so it should make sense that movement becomes abnormal in these individuals. From the table, we can see that basal ganglia atrophy leads to chorea and athetosis. Chorea is characterized by involuntary abrupt movements of the face, trunk, and limbs, and athetosis is characterized by writhing hand movements. Because the brain is affected, it should be no surprise that this disease can also cause psychiatric changes, such as aggression, depression, and dementia. We talk about this idea more in neurology, but atrophy of the brain can cause the ventricles to expand, which results in a condition known as hydrocephalus ex vacuo. Decreased concentrations of acetylcholine and GABA, as well as elevated dopamine levels in the brain, are associated with this condition. Because CAG codes for glutamine and Huntington disease is caused by CAG repeats, this disorder results in excess glutamine in the brain, which is an excitatory neurotransmitter. Therefore, this can result in glutamine excitotoxicity. Finally, the CAG repeat can be remembered by recalling a few facts about the disease. So, caudate, acetylcholine, and GABA. Notice that the first letter in each of these words corresponds to the trinucleotide repeat CAG. Friedreich ataxia is an autosomal recessive condition caused by GAA repeats. The repeats occur in the FXN gene, which codes for the frataxin protein. This is an iron-binding protein, and defects in the protein result in mitochondrial dysfunction. Mitochondria are heavily present in the brain, pancreas, and heart, which explains many of the symptoms, including neurological deficits, diabetes mellitus, hypertrophic cardiomyopathy, and kyphoscoliosis during childhood, as well as feet deformities. The neurological deficits can be explained by understanding the various pathways that are affected. However, perhaps the most important neurological symptom to remember is ataxia, hence the name Friedreich ataxia. The trinucleotide repeat can be remembered with the mnemonic gait is ataxic. Notice that the GAA in gait corresponds with the GAA repeat. Myotonic dystrophy is an autosomal dominant condition caused by CTG repeats. The repeats occur in the DMPK gene, which results in myotonin protein kinase dysfunction. Myotonic dystrophy is called myotonic because patients have prolonged muscle contractions or myotonia. Other findings include muscle weakness, cataracts, gonadal atrophy, frontal balding, and arrhythmias. The trinucleotide repeat can be remembered by recalling a few facts about the disease, so cataracts, tired muscles, and gonads. Notice that the first letter in each of these words corresponds to the trinucleotide repeat CTG. Fragile X syndrome is an X-linked dominant condition caused by CGG repeats. The repeats occur in the FMR1 gene, which results in hypermethylation and silencing of the gene. I discussed DNA methylation and silencing more in molecular biology, so we're not going to talk about that more right now. Symptoms include a long face, large jaw, large ears, macroorchidism, neuropsychiatric changes, mitral valve prolapse, and joint hyperlaxity. Joint hyperlaxity is extremely important to keep in mind because this can easily be confused with Ehlers-Danlos syndrome. Fragile X syndrome is the most common cause of inherited intellectual disability. Inherited here is a key word because Down syndrome is the most common cause of intellectual disability, but this is usually due to non-disjunction, so it's not really an inherited condition that follows a mode of inheritance. Whereas Fragile X syndrome is an X-linked dominant condition. The trinucleotide repeat can be remembered by recalling a few facts about the disease, so chin, gonads, and gene silencing. Notice that the first letter in each of these words corresponds to the trinucleotide repeat, CGG. Okay, let's do a question. A 31-year-old male presents to the physician due to a 3-month history of depression and memory loss. An MRI of the brain reveals atrophy of the caudate. His father developed similar symptoms, but at a much older age. What genetic principle explains these findings? Hopefully from the question stem, you notice that this patient has depression, memory loss, and an MRI that shows atrophy of the caudate. These are classic findings in Huntington disease. The development of symptoms at a much younger age compared to his father can be explained by anticipation. So again, anticipation is the tendency of a disease to occur earlier or to be more severe in succeeding generations. Okay, let's do another question. A 7-year-old boy with a history of anxiety presents to the physician for heart palpitations. On exam, he has a murmur heard over the left sternal border. He also has a long face and large testes. PCR of the affected gene would most likely reveal what trinucleotide repeat? A, GAA, B, CTG, C, CAG, or D, CGG. So this boy has anxiety, a cardiac murmur, a long face, and large testes. Hopefully you remembered that these findings are consistent with Fragile X syndrome. Fragile X syndrome is caused by CGG repeats, so the answer is D. From the table, you can see the repeats right here, as well as the mnemonics over here. Hopefully you remembered the mnemonic chin, gonads, and gene silencing. As we can see, this corresponds with the repeat CGG.